Eriku, and it's a great honor and privilege for me to introduce Chris Glass from the Cellular and Molecular Medicine Department at UCSD as today's Shiley-Marcos ADRC Neurodegeneration Seminar speaker, and also co-sponsored by the T32 Training Grant in Alzheimer's disease. One of the pleasures of doing the introduction is that I get the chance to read the CV of the speakers, especially in this case of an outstanding researcher. It's entirely justified to say that Chris Glass is now one of UCSD's trophy faculty. Indeed, UCSD should be very proud to have trained Chris and to have him stay on in our faculty for essentially his whole career. With the exception of his undergraduate degree from Berkeley and 2 years at Brigham and Women's Hospital in Boston for internal medicine residency, Chris has been at UCSD the entire time. He received his MD PhD from UCSD in 1984, doing his thesis work with Daniel Steinberg, one of the pioneers in lipid and lipoprotein biology. Chris then left for Boston for 2 years after graduating, but returned to UCSD for his endocrinology fellowship, this time in Jeff Rosenfeld's lab, working on thyroid hormone receptor biology. From 1989 on, he started first as assistant professor and progressing to full professor in 1999, as well as being named the University of California Distinguished Professor in 2018. His work has been focused primarily on understanding transcriptional control, co-activators, and co-repressors in the development and function of macrophages in health and disease. But important for today is his more recent focus on macrophage activation in inflammatory conditions, and also in macrophage activation in neurodegeneration and Alzheimer's disease. I'm personally excited when distinguished researchers such as Chris bring their expertise and skill set to look at Alzheimer's disease and neurodegeneration. I think this bodes well for our field, as we surely need more basic understanding of the pathophysiology of AD. Needless to say, Chris has received many honors, and I will mention just two. His membership in the National Academy of Medicine, the Institute of Medicine honor, and in 2017, election to the National Academy of Sciences. Finally, this is one of those rare times when I stare in admiration, as Chris is, as I said, a member of the National Academy, but so are his two former mentors, that's Dan Steinberg and Jeff Rosenfeld. It's a very uncommon achievement when both mentor and mentee are elected to the National Academy. However, the first time I ever saw this combination was also from UCSD. I was in a study section when a grant from Ted Bullock was being reviewed about 20 years ago. For those who are too young to know, Ted is truly a pioneer in the world of neuroscience, working in invertebrate neurophysiology from his lab at SIO. In fact, that Scripps Institute of Oceanography, before UCSD was even founded. I had to leave the room during the review of his grant, which was submitted when he was already in his 80s. When the review was finished and I returned to the room was abuzz over a statement in his bio sketch, where he said he is a member of the National Academy of Sciences, but so are also 4 of his former graduate students and postdocs. So here, history repeats itself with Chris and his own research mentors as well. So without further ado, here's Chris Glass giving his talk today entitled Decoding Microglia Phenotypes in Neurodegenerative Disease. So thanks very much, Chris. Looking forward to your talk.
Well, Eddie, that was probably the warmest, most generous introduction I've ever had. So thank you very much for that. And you know, it's an honor, but it's also a challenge to be presenting on the home field because you've got members of the lab attending. I guess they can attend now anywhere with Zoom, but this is always a challenge because we have a number of very interesting stories that are in the pipeline right now, and I can't tell all of them. And because this is an Alzheimer's disease focused seminar series, I'm going to just select the studies that are ongoing now that are most relevant to that interest. And these fall within our general efforts to try and decode not only microglia phenotypes, but also macrophage phenotypes throughout the body. And so this talk will be focused on microglia and neurodegenerative diseases. And I think these cells are probably familiar to almost everybody online. I'll give them a short introduction. They were discovered by Uriel Ortega and reported in a very amazing series of papers that came out between 1919 and 1921. He was a sort of a trainee of Ramon y Cajal, and what he did was to apply new staining methods, and in particular the Golgi-Ortega method, to allow him to see what was initially called the third element, and then termed the microglia population of the brain. And his drawings of these cells, sort of follow in the tradition of Ramon y Cajal, and here's a drawing of his vision of microglia living in the cortex. And of course, we now have many other methods to visualize these cells, and one of them is the ability to transgenically express green fluorescent protein in them very specifically. And so here's a picture from Axel Nimmerjohn, who's now one of our colleagues over at the Salk Institute, that he took now more than 15 years ago, showing striking similarities. These cells we now appreciate are derived from yolk sac erythromyeloid progenitor cells, very early in development, really as soon as a vascular system invades the head, they take up residence within the brain, the developing brain. They're a self-renewing population that is independent of hematopoietic stem cells. We know that they're phenotypically diverse, that like all macrophages, they're sentinels of infection and injury. They have a number of properties that are very typical of macrophages, such as their ability to phagocytose apoptotic cells. And in the brain, they're important cells in the clearance of protein aggregates, such as amyloid. They play roles in the refinement and monitoring of synapses. They modulate neurogenesis and blood vessel formation. And what has attracted tremendous attention to these cells, really based mostly on the findings of genome-wide association studies 6 to 8 years ago, is their genetic linkage to neurodegenerative and behavioral diseases, in particular Alzheimer's disease. So we know about this association, and there are a number of general questions that are being pursued very intensively by a large number of neuroscientists and neuroimmunologists. And these include asking the question of what are the functions of these molecular distinct microglia under homeostatic and pathological conditions. We're very interested in the mechanisms that drive these phenotypes. We're interested in how these mechanisms are affected by genetic variation and environmental perturbations. And ultimately, we're interested in whether in the idea that knowledge of these mechanisms could be used to develop new treatments for prevention, or new approaches for the prevention and treatment of neurodegenerative diseases. Now, there are a lot of ways to approach some of these general questions. The approach that we primarily use was alluded to by Eddie in his introduction. We're a lab that's focused on trying to understand transcriptional mechanisms that regulate gene expression and thereby determine the potential of a cell to adopt a particular phenotype. And so this focuses our attention on the regulatory elements of DNA, which are outlined here in this slide. At the bottom is a depiction of promoters. These are the obligatory start sites for messenger RNAs. They tend to be regulated by transcription factors that are relatively general, that don't necessarily show a high degree of cell specificity. And because of that, while they're able to set up an active transcriptional start site, promoters generally don't provide the information that's necessary for a cell-specific developmental program of regulation, or the ability of a cell to respond to signals in a cell-specific manner. And that function is primarily provided by enhancers. These are regions of the genome that can be upstream of the gene, within introns of genes, downstream of genes, and they turn out to be the major places in the genome that are occupied by what we refer to as lineage determining transcription factors, or LDTFs. So these are the transcription factors that are essential for driving specific cell fates. And in microglia, I'll give you a little bit more information as to what these are, but basically these factors function to open up enhancers throughout the genome in a very cell-specific manner, and provide sites of interaction for signal-dependent transcription factors that enable the cell to respond to internal and external signals. And so a major effort in our laboratory is to try and understand how these enhancers are selected, how they are influenced by the internal and external environmental factors, and how their functions can be perturbed either by environmental perturbations or genetic perturbations. Now, we can quantitatively measure many of the features on this diagram. We can measure RNA levels very precisely using RNA sequencing and single-cell RNA sequencing technologies. And those methods really give us sort of a molecular phenotype of a particular cell. But in order to understand how the cell turns on a specific program of gene expression, then we want to sort of look under the hood and understand how these transcriptional regulatory elements are set up. And to do that, we have a number of other types of assays. One is an assay to measure open regions of chromatin. We can use an assay called a TAC-seq to do that. And to give you an example of what that data looks like, I'm showing you a region of the genome in the vicinity of the BIN1 gene. So in this genome browser image, the promoter of BIN1 is shown here, and the gene is being transcribed from right to left. This is the first exon where the promoter of BIN1 is, and this little arrowhead dashed line represents the first intron. And you can see that there's an ATAC-seq signal here. This tells us that there's open chromatin of BIN1, and this experiment was carried out using human microglia. And in addition to this open region at the promoter, you can see upstream these additional regions that are open. And so these would potentially be enhancers. And so that then leads us to asking, well, okay, is there evidence for enhancer activity? And one of the key methods that we use to look at that is a chromatin immunoprecipitation, or CHIP, for histone modifications. And the example that I'm going to provide here is a CHIP for a modification or acetylation of histone H3 lysine 27. So when histones are acetylated at this position, that's a mark that's very tightly linked to promoter and enhancer activity. And in these cells here, these microglia cells, you can see that the histones in the vicinity of the BIN1 promoter are acetylated, but they're also acetylated upstream at these regions of open chromatin, which we would take as a sign that these regions of open chromatin have the properties of active enhancers. Now, to study how those are selected from the genome, we can perform CHIP sequencing for transcription factors. And here's an example of a CHIP for the lineage determining factor PU.1 required for microglia development. You can see that it's binding at the promoter and at these upstream regions. And so this would provide evidence that PU1 is playing a role in establishing these enhancer elements and allowing them to become active. Now, one of the challenges in interpreting these enhancer maps is knowing the genes that the enhancers are connected to, because enhancers don't necessarily regulate the nearest gene. And to look at that, we can use an assay called PlaqueSeq, which allows us to study interactions between enhancers and promoters. And an example of that in the vicinity, the BIN1 gene is provided here. And these little loops tell us that these regions of DNA are actually close to the BIN1 promoter in three-dimensional space. So with this general background into the approach, I'm going to first take you back to 2017, which was our first paper that allowed us to begin to make some statements about human microglia gene expression and the mechanisms that are responsible for driving the human microglia phenotype. And these experiments were made possible by Nicole Kufal, who is also one of our MSTP graduates and is carrying out her career here. She's a pediatric intensive care physician, and also, as you'll see, a neuroscientist and a specialist in iPSC technology. Nicole, through her interactions with the surgical colleagues, Michael Levy and David Gondo, enabled us to collect discarded samples from pediatric patients undergoing surgery for a variety of brain diseases, primarily tumors and epilepsy. And by carefully collecting these samples and doing our best to try and pick regions of the brain that were not affected by the underlying disease, we were able to isolate microglia to study both ex vivo and in vitro, and to perform transcriptomic and epigenetic analyses. And so this led to what at the time was the first transcriptome of human microglia. And it gave us the first indication of what genes were specific to microglia versus to the rest of the brain. And taking highly expressed genes, we could see the immune signature of these cells and get an appreciation for how different genes exhibited variable expression across individuals. One of the key experiments that David Gosselin carried out using these cells was to, in addition to studying their expression immediately after they were removed from the brain in this ex vivo context, was to put the microglia into culture and to watch how the gene expression changed in these cells as a function of time. And what this heat map is showing is that about 800 genes, when the cells are put into culture, show dramatic upregulation, and about 1,200 genes show very dramatic and very rapid downregulation. And this loss of gene expression is very highly enriched in the set of genes that we think of as the genes that provide microglia with their identity. And what this experiment told us was that the ability of the microglia to establish their state of gene expression is very dependent on the brain environment. And it actually requires constant reinforcement of these brain environmental factors in order to retain this transcriptomic state. Now, interestingly, when we looked at the genes that are decreased in vitro, we saw that there's a very strong overlap with genes that had been defined as being upregulated when the hematopoietic progenitor cell migrated from the yolk sac into the brain early in development. And so from this analysis, we concluded that a lot of these downregulated genes were indeed not just an artifact of tissue culture, but really indeed reflected genes that required brain environmental signals that were no longer there. And just to give you an example of this, one of the genes that we're studying most intensively is a transcription factor called SALL-like-1, and at some point I will come and tell you a lot more about that. This is work being led by Bethany Fixon in the lab, one of our MSTP students. This transcription factor is absolutely essential for the maintenance of microglia identity, and its expression falls dramatically in vitro. And this occurs in concordance with a dramatic loss of activity of what we believe, and actually now Bethany's actually proven, is the SAL1 super enhancer. This region of genomic regulatory DNA loses a tremendous amount of its activation signal in the in vitro environment. And so from using a combination of the open chromatin, the histone acetylation, and the changes in gene expression, David, working with an informatics student, Dylan Scola, and Nicole, were able to put together a model for how microglia develop from a progenitor cell in the yolk sac to a microglia in the brain. And the basic idea is that there's a core set of transcription factors that are common to basically all macrophages within the body that provide information needed for core macrophage functions. And as these cells migrate into distinct environments, such as the brain, the cells now become under the influence of those environmental signals, which have a number of consequences. They can change the activity states of existing factors, such as the SMADs. They can induce the expression of receptors, such as cell-like 1. And this combination of events will then allow them to form collaborative interactions with each other and core macrophage transcription factors to open up enhancers that drive microglia-specific functions. And these processes can, of course, be affected by changes in the brain environment, such as due to infection or toxins or protein aggregates or what you had for breakfast that morning, as well as perturbations of the genome. So there are, for example, well-known variants in the regulatory elements for Q1 that alter its expression. MEFs can be altered by genetic variants, as well as genetic variants that hit a variety of enhancers. And so a major interest then is how these combinations of environmental and genetic factors affect pathogenic gene expression. Now, as I mentioned, a lot of interest in microglia was sparked by the initial findings of genome-wide association studies. And I think most people that have looked at the results of these studies are aware that for the most part, the variants that are identified in these studies that are associated with risk are not associated with changes in protein coding regions. Some are, and some of those are very important. So for example, ApoE here, the most significant, this is where the most significant risk alleles lie. These variants do indeed change the amino acid sequence of ApoE to generate ApoE2 or E3 or E4. But most of the genes on this list are on this list not because genetic variation has altered their protein sequence, but because genetic variation is close to that gene. And I'll get back to this in a minute. But one of the difficulties here is that because this is non-coding genetic variation, it's very difficult to interpret. So it's not immediately clear what gene these variants might affect or what cell type these variants might have activity in. And this is where the studies of the cell-specific transcriptomes and the epigenetic or enhancer landscapes can be very useful. And going back to the original paper from David Gosselin and Dylan Scola and Nicole Kufal, we were able to take the variants that were at that time associated with risk of Alzheimer's disease, line them up with the nearest gene, and then ask, where are they expressed? Are they most highly expressed in microglia, or are they most highly expressed in the brain? And our finding, which was really consistent with the emergent recognition of of microglia genes being somehow linked to Alzheimer's disease, 31 of these loci were near genes that were most highly expressed in microglia compared to the cortex. Now, because we only had at that point a transcriptional map or an enhancer map for microglia, we really couldn't put this into context. And so one of the next major efforts was led by Alexei Knott, who had the hypothesis that if we had a cell-specific enhancer atlas for the other cell types of the brain, that would facilitate our interpretation of disease risk alleles. And this effort, I should say, was very significantly supported by the ADRC. So Alexei got one of the ADRC pilot feasibility grants, and that helped him to develop a method for fluorescence activated cell sorting, where he was able to take the brain tissue, isolate nuclei, stain them with antibodies for specific cell types, and by using fluorescence activated nuclear sorting, then sort the nuclei into microglia, astrocytes, neurons, and oligodendrocytes. And then once he did that, he could take each population of nuclei and run them through the genomic assays that I've described. And doing that, what he found was, this is really not too surprising, the promoters in these four cell types were largely overlapping. About 10,000 promoters were identified that were common to each of these four cell types. There were cell-specific promoters, such as the 913 identified in microglia. But if you look over here at this sort of bizarre Chow-Ruski version of a Venn diagram, what you can see is that the enhancers of each of these cell types were much, much more cell-specific. So we identified about 24,000 enhancers in microglia that were cell type specific, 92,000 in neurons, 27,000 in oligodendrocytes, and 25,000 in astrocytes. And this data set allowed Alexei, working with Inga Holtman, a geneticist, and again, Nicole, to align these cell-specific enhancer elements with the genome, with the risk alleles or alleles associated with various traits defined by genome-wide association studies. And here we observed a striking enrichment of risk alleles selectively in microglia-specific enhancers, whereas for other traits, such as psychiatric traits, these risk alleles were primarily associated with neuronal enhancers. Now, this was very interesting. It clearly set up a hypothesis that risk alleles that were associated with microglia-specific enhancers were probably acting in microglia, but this is a difficult problem to actually validate due to the nature of working on human cells. And just to give you an example of what this would look like, so returning now to bin1, which I showed you at the beginning, here's the promoter. Now we see the whole bin1 gene, and the enhancer is here, and the most significant risk allele from the GWAS studies is right in the middle of this yellow bar. And what I'm showing you below here are the histone acetylation tracks for microglia, neurons, oligodendrocytes, and astrocytes. And what you can see is that this risk allele is residing in a region of the genome that really only looks like it has enhancer activity in microglia and not in neurons, oligodendrocytes, or astrocytes. Now, to look at whether this was really a risk allele that would be associated with the microglia-specific enhancer, Nicole deleted this region, about 300 base pairs, from iPSCs, and then differentiated them into astrocytes, microglia, and neurons. Now, bin1 is expressed in the iPSCs and in each of these cell types. And in fact, most work on on BIN1 and its relationship to Alzheimer's disease has actually been in neurons. Now, when she differentiates the cells into neurons, this deletion has no impact on the expression of BIN1 in those cells. And the same is true when the cells are differentiated into astrocytes. But when the cells are differentiated into microglia, the BIN1 enhancer deletion has a very dramatic impact on BIN1 expression. And so this would be, I think, a pretty strong piece of evidence that the risk alleles in this location of the genome are associated with risk of Alzheimer's disease because they affect the expression of BIN1 in microglia. And that is a problem that we're continuing to advance with Nicole. Anna Warden is a postdoctoral fellow that's joint between Nicole and I, and Anna is pursuing this direction. I'd like to use the rest of my time to tell you about a relatively new direction that is unpublished and that we're making, I guess, I could say enough progress to be able to talk about it, that has to do with a relatively recently identified microglia phenotype. And these are referred to as the disease-associated microglia, or DAMs. They were defined based on a single-cell RNA-seq experiment using mice, in which microglia, actually all of the hematopoietic cells of the brain, were subjected to single-cell analysis, either in a wild-type mouse or in a mouse, the so-called 5X FAD mouse, which is a model of amyloid. And this work was carried out by Ido Amit's laboratory. And I should say Oleg Botovsky also carried out parallel experiments, which allowed him to come to similar conclusions. And what the major finding is that a small fraction of the microglia in the 5X FAD mouse brain had a distinct phenotype that could be appreciated by the single cell analysis. And in looking at the distribution of differentially expressed genes, there were a couple hundred genes that were specific to this disease-associated cluster of cells, and that could be used as a DAM, so-called DAM signature. Now, this finding was very interesting because among the most highly expressed genes of the DAMs was TREM2. And as I'll show you in a minute, this phenotype is TREM2 dependent. And that discovery was made in the context of prior work that had looked at the impact of deleting TREM2 in the 5X FAD mouse model. And so what was found in this case, work with that was led primarily by Marco Colonna, but other laboratories have also seen similar findings, is that if you eliminate TREM2 from microglia in the context of this 5X FAD model, what you observe is that the microglia, which normally are able to migrate to plaques, and these plaques are fairly compact, in the TREM2 knockout, the microglia are no longer able to effectively home to the plaque, and the size of the plaque spread. And so this it was consistent with the idea that the DAM phenotype is dependent on TREM2, and that this ability of TREM2 to enable microglia to home to plaques and potentially keep them compact was possibly explaining some of the protective functions of TREM2 suggested by genetics. Now, this finding in the brain turned out to have counterparts in other tissues. And I'm just bringing this up not only because we're interested in macrophages in other tissues of the body, but because we think that understanding what TREM2 is doing in a variety of different macrophage populations will help inform us as to what it's doing in the brain. The AMIT laboratory in 2019 reported a TREM2 dependent population of macrophages that surround apoptotic or necrotic adipocytes in obese adipose tissue. These were full of lipid, and he referred to these as lambs. And then in the same year, a group studying cirrhosis in the human liver identified another TREM2 population of macrophages that were called SAMs that decorate collagen scars. And these phenotypes are interesting. They have a number of common features. They're all associated with focal areas of tissue pathology. They appear to play protective roles. They're dependent on TREM2. They contain intracellular lipid droplets, and they exhibit partially overlapping gene expression signatures, which suggests that they have at least partially overlapping functions. These include genes such as CD9 and NPC2. Now, I think one important point in the description of these cells is that they don't really seem to correlate with previous classifications of macrophages or microglia into M1 or M2 phenotypes. Now, what was shown in Ito Amit's paper was that if you looked at the microglia in the FAD model in a TREM2 knockout, that you did not get the DAM phenotype. You got a partial transition of microglia towards a DAM phenotype, but you didn't get all the way there. And so this set up the concept of a TREM2 independent phase and a TREM2 dependent phase of the DAM phenotype. And so here's where we come in. So we're very interested in this phenotype and how to possibly modulate it and what it actually means with respect to Alzheimer's disease phenotypes. TREM2 was originally cloned by Marco Colonna approximately 20 years ago. So this has been a molecule that's been around for a long time. It's a pattern recognition receptor, but not, as far as we know, not really a pattern recognition receptor for pathogens. It seems to mostly be dedicated to molecular patterns that are associated with cell death or tissue injury. When it is ligated, it forms a complex with DAP10 or DAP12 that signals through Syk and PI3 kinase. Now, what's remarkable is that even though this receptor has been around for so long and that it is now known to be required for this DAM phenotype, we have very little knowledge of what the signals, receptors, or transduction pathways are that allow this gene to be turned on. And similarly, although we know this couples to Syk and PI3 kinase, what's below those signal transduction pathways is really almost a complete black box. We know almost nothing about the transcription factors that mediate TREM2 dependent functions. And so in the last few minutes, I'll just outline our a general approach of trying to use what we refer to as dynamic enhancer landscapes as a strategy for uncovering the transcription factors and upstream signaling pathways that drive this particular state. And this is applied to understanding DAMs, but you can think of this as a general approach for looking at any change in state of a cell type. And so the idea is that environmental signals, of which there are many, and the most important ones are really unknown, will interact with TREM2 and lead to signaling. And this will change the activities or expression levels of transcription factors that drive transitions of enhancers from closed or inactive to open and active. And as I mentioned to you, we can quantitatively measure these changes using a combination of ATAC-seq and ChIP-seq for H3K27. So by comparing the enhancers that have been opened and activated by a process such as living in the 5X FAD brain to the enhancers under a control condition, we can carry out motif enrichment analysis, and this will yield motifs that we hypothesize are the binding sites for the transcription factors that are driving these changes. And then once we can identify these factors, then we can begin to work our way upstream and ask how they become activated in response to the initial signal. And so to do this, we have collaborated with Matthew Blurton-Jones' laboratory. I think many of you are familiar with Matt. He actually trained here at UCSD in the Department of Neuroscience, I think with Mark Jasitsky, and then has had a great career at UC Irvine. And one of the most significant things that he's done over the last couple of years, one was to develop very robust methods for converting iPSCs into microglia, but he then developed a system where he would introduce hematopoietic progenitor cells derived from iPSCs into a immunodeficient CSF1 humanized mouse that would allow these cells to engraft in the brain and differentiate into microglia. And this model, it doesn't completely solve the problem that I told you about taking microglia and putting them into a culture dish, but it mostly solves the problem of generating an in vivo human microglia phenotype. And that is accomplished by putting these cells into the humanized mouse brain. And the key factor that is humanized in these mice is CSF1, which is a major growth factor for microglia. And so these mice can subsequently be crossed into the 5X FAD background. And when this is done and single cell RNA sequencing analysis performed, Matt, and I should actually say this is work done by Johnny Hasselman, a graduate student in Matt's laboratory. So when Johnny carried out single cell RNA sequencing of these cells, he basically recapitulated the findings of Ido Amit's laboratory, identifying a population, in this case, human microglia in the 5X FAD brain that had this disease-associated microglia signature, as well as populations that were enriched for MHC class II molecules and signs of interferon signaling. So these, in addition to the so-called homeostatic populations. From the single cell analysis, Johnny was able to get markers that allowed him to sort each of these cell types. And then Alexei carried out a TAC-seq on each of these populations in order to try and identify so-called disease-associated microglia-specific enhancers. And the analysis of this data was carried out by Eric Kaufman, who's a bioinformatics graduate student in the laboratory. And Eric was able to identify disease-associated microglia-specific enhancers in these cells. And the motif analysis gave us quite a few significant motifs. And the top motif on the list here is a transcription factor. Well, the motif is recognized by a transcription factor called EGR2. This is a factor that's been known to play important roles actually in a variety of processes and other types of macrophages. It's downstream of IL-4 and wound repair in other contexts. Motifs for the TFEB family, these are transcription factors that are known to have important roles in autophagy, phagocytosis, lysosomal function, and of course, very interesting with respect to the DAM phenotype. CBP, a core microglia lineage determining factor, ATF3, a member of the AP1 family, and an NF-kappa B motif. And so this gives us actually a lot to work from. This is a very interesting list, suggests that these are either upstream or downstream of TREM2 signaling. And so I'll just focus mostly here on EGR2 as an example. If we actually look at the gene expression pattern in the DAMs in this model, EGR2 is upregulated. And so now we're faced with this question of how do we validate any of these findings? And it's really analogous to the problem that I mentioned earlier about, how do you validate a risk allele or microglia specific enhancer? And so here, again, we've turned to an in vitro iPSC-derived microglia model system. And Matt had recently shown that he could quantify TREM2 signaling in human iPS-derived microglia by basically setting up either wild-type iPSC-derived microglia or TREM2 knockout microglia, and then activating these cells with an antibody that binds to TREM2 and leads to increased signaling. So as part of a collaboration with the Cure Alzheimer's Foundation, we've worked together, and Matt's laboratory did, I think, what was sort of an industrial scale experiment where they grew massive amounts of iPSC-derived microglia from 2 different lines that were either wild type or isogenic for a TREM2 deletion. And then these cells were plated in the absence or the presence of this activating antibody for TREM2. And then we took a guess, this is really a gamble as to when the best time to look for changes in the enhancer landscape would be. And based on the concept that TREM2 would signal through a cascade involving PI3 kinase, we looked at a very short time interval, 2 hours. And Johannes Skoletsky, working with Sidney O'Brien and Chris Balick, are now in the process of undertaking a massive ChIP-seq and attack-seq approach to define the TREM2 dependent enhancers. And so what they're finding, this is just very new data. We're finding that in the attack-seq data, there are differential enhancers that show distinct amounts of open chromatin, and the top motif on the list that comes out of this is EGR2. And similarly, in looking at the acetylated enhancers, they're finding that the top motif is a motif for P1 or SPI-B, but the second motif is a motif for EGR2, and now we're also seeing the NF-kappa B and ATF motifs that I had alluded to in the transplanted model. So this data, in combination with other types of data, not only in microglia, but in other tissue-resident macrophages, are beginning to give us what I would call a working model for mechanisms that drive the DAM, SAM, LAM phenotypes in chronic disease. And I mentioned that there's a TREM2 independent phase of DAMs, and I think that's going to be true for the SAMs and the LAMs. And what it appears is that the microglia are not and immediately, well, I guess what I should say is what these phenotypes really reflect is a 2-stage process, where the initial stage is for the macrophage or the microglia to detect tissue damage of one sort or another. And this initial detection of tissue damage is what represents the TREM2 independent phase, and is the process by which TREM2 is activated. And in studies that I don't have time to discuss, we think that this is at least partially related to activation of ATF3 possibly the liver X receptors, and possibly the MITF-TFE family. And this leads to upregulation of TREM2, which is one of the defining genes of the DAMs, as well as TREM2 independent genes. Now, once you get TREM2 on and it sees its ligands, it can signal through this PI3 kinase dependent pathway to activate other transcription factors. We think EGR2 is one of these. ATF3, TFEs, NFATs, and NF-kappa B may also be downstream, and that these will then collaborate to turn on target genes. Now, this model, I think, may be actually a lot more complex, and this really gets to one of what I think are the key questions about microglia in the brain and their relationship to Alzheimer's disease. So we've defined this disease-associated microglia phenotype, and that is a phenotype that, based on the loss of function of TREM2, is protective in the brain. But we also think that microglia are drivers of inflammation and tissue pathology. And so I think there's really, there are two sort of general ways to think about it. One would be that the DAMs are both good and bad, and that the DAM phenotype, that the downstream processes that are associated with protection dominate. And so that's what this model is showing, and that is that TREM2 is activating these combinations of factors, and that these factors can then go on to activate target genes that are both protective and pathogenic. So if that's the way this works, then we want to sort this out. An alternative is that there are other macrophage or other microglia phenotypes in the brain that are not DAMs, but are microglia that are dedicated to tissue pathology. But as yet, there really isn't any strong evidence that that is the case. And so that's where we are right now. I think the next steps on this project will be to intersect some of these DAM-specific enhancers with GWAS risk variants. This may lead us to discovering new enhancers that may mediate effects of non-coding alleles that we couldn't pick up using our previous samples. But now we really want to try and really validate whether EGR2 is a downstream target of TREM2, looking by ChIP-seq for it, looking at it in DAMs and possibly human AD brain samples, and to look at the consequences of loss of function of EGR2. And then beyond that, further exploring other transcription factors that show enriched motifs in DAM-specific enhancers, such as ATF3 or the MITF-TFE family. So the main take-home conclusions for really the last part of the talk are that dynamic enhancer landscapes enable the inference of transcription factors that drive microglia identity and function. Many Alzheimer's disease risk alleles reside in microglia-specific enhancers and are likely to exert phenotypic effects in microglia. There are major knowledge gaps remaining in defining the brain environmental signals and downstream pathways necessary for the development of homeostatic and pathogenic microglia phenotypes, and that disease-associated enhancers implicate members of the EGR, MITF, and ATF families of transcription factors as drivers of the DAM phenotype. And so with that, I've mentioned many of the people that have been involved. The alumni here had a big role in all of the published work that I told you about. And as I mentioned, Alexi was supported directly by our ADRC, and is very grateful for that. Almost everything that I've told you about has been hugely influenced by Nicole Kufal, and she has been an absolutely great collaborator. Bing Ren has helped us with some of the plaque-seq data. We couldn't do any of the work on human subjects without our surgical colleagues, Michael Levy and David Gonda. I've mentioned Matt and Johnny. Amanda McQuaid was the graduate student in Matt's lab who set up that massive iPSC-based experiment. And then in the newer work, again, shout out for Johannes. I think many of you know Johannes. working with Eric and Chris Balick and Sydney O'Brien on this most recent experiment. I mentioned Anna on the BIN1 project, and I would love at some point to tell you about the work that Claudia Hahn and Bethany Fixon is doing. Maybe we'll do that next time, and then mention Martinez being involved in all the sorting projects. So with that, thank you very much for your attention. I'd be happy to take any questions.